Okay, great. So I'm very pleased today of introducing our speaker, which is Alex Mogilner. Alex has had different positions in Yekaterinburg, Manitoba, British Columbia, and California in the past. And as of 2014, he's a professor at the Courant Institute and also at the Department of Biology at the NYU. His research interests include the dynamics of molecular motors, microtubules, cell motility, cell division, and many other amusing things that are taking place in our cells. In particular, he has made crucial contributions to our present understanding of the mitotic spindle, and I bet we will learn something about this precisely today. So, Alex, we are all ears.
Thank you very much for a general and very generous introduction. So I will tell you today about some quantitative aspects of mitotic spindle assembly. This is one of my favorite areas in science, and lately we have been involved in some very exciting new developments in this area. I want to share them with you. So the people who were involved in this project over the past few years are shown on this slide. Chris Miles is my postdoc at Courant. He is responsible for all the modeling and data analysis in the second part of the talk. For the short first part of the talk, I'm collaborating with a group from University of Massachusetts, which is shown at the bottom of the slide. And the experimental results, there'll be plenty of them, are obtained in the lab of Alexei Kodjakov, who is in Department of Health in Albany, New York. It's a very unusual place for scientific lab. So these days they do very little experiment. They heavily involved in making and distributing one of the COVID vaccines. They're the heroes. And Fiorana was a postdoc who actually obtained those experimental results. So I wanted to start with this fragment from Diego Rivera's mural, MNat Crossroads. You can actually see this mural in Mexico City in one of the magnificent buildings there. And the reason I put it there is that at the very center of this mural, there's this little painting of mitotic spindle in metaphase. And it's quite amazing that that was done 100 years ago, yet all the essential details are captured very, very faithfully. Since then, the knowledge increased orders of magnitude, but still the general view is amazingly similar to this stunning micrograph of actual spindle. So there are many interesting questions about mitotic spindle, but one of the main ones is how does the spindle actually assemble? Here's the kind of watered down, very simplified view. In the beginning, prior to mitosis, the cell has two centrosomes that are tiny complex organelles, but for the purposes of today's talk, they're just anchoring points for the so-called microtubules. So microtubules are these linear polymers that have two very different ends. They have minus ends that are anchored in the centrosomes and static, and plus ends that are very dynamic, and I will discuss the dynamics in the next slide. So together with microtubules, centrosomes organize these asters, right? We have these two microtubule asters. It's kind of the sun with rays of the sun coming out. And we have chromosomes. Now, prior to division, chromosome is two sister chromatids, right? Two identical genetic information units. They are glued together until the end of the metaphase, after which the sisters are pulled separately into and oppositely into future daughter cells. But we're not going to get this far in this talk. So at the so-called centromere, the middle part of each sister chromatid, there's a complex protein machine called Kinetochore. And it's very complex. There are tens of labs working on the structure and function and dynamics of the kinetochore. Again, for most of today's talk, I will not go into this very delicate process on the kinetochore. I'll just say that this is, in a way, a certain glue for the microtubules. So microtubules, when they reach the kinetochores, they get glued to it. Later in the talk, it will be clear that that's not exactly true. So the early view of the spindle, there were these two opposing concepts, how the spindle assembles. One was that these two asters, microtubules, somehow connect to the kinetochores. After the connection are made, here you are, you get your spindle. Two centrosomes at the pole, chromosomes at the equator. So you can imagine that this is Earth with south and north pole, and the equator with the chromosomes. Opposing view was that centrosomes are not actually important. And this opposing view comes from observations that some spindles, for example, meiotic spindles, they can get organized without the centrosomes. And this view is that microtubules are actually coming from the kinetochores or somehow emerging in the vicinity of the chromosomes, and then somehow the minor ends of the microtubules get focused and you have your spindle. Okay. So you can imagine that there are many detailed questions that challenge both views. So gradually, there's this emerging recognition that reality is a hybrid of those two processes. And that's kind of philosophically the pervading theme in biology these days, that the extreme views normally means that in the end, there will be some convergence and parts of both processes will be true. So that's what I wanted to talk to you about today. Now, I wanted also to mention that there are three interesting features that people thought is associated with this assembly of the spindle. Actually, four features. One is the spindle self-assembles, not just assembles. So there's no central brain in the cell that tells the cell, okay, assemble this spindle there. Somehow it just self-assembles from these parts that are floating around in the cytoplasm. Another is that the spindle assembles stochastically. It will be clear from the next slide what it means. But the two features are unquestionable. That's observed again and again. The spindle assembles rapidly. It just takes minutes in most of the generic animal cells. And it assembles very accurately. Again, from the next slide, it will be clear how it happens. So the first physical and mathematical idea on how the spindle assembles came from the work about like 35 years ago that started with Michelson and Kirchner, two demigods of modern cell biology, who in the middle of the 80s discovered the process called the microtubule dynamic instability. So they discovered that microtubules rapidly and repeatedly grow and shrink, grow and shrink. Okay, and those cycles of growth and shrinking were characterized in mathematical detail. Turns out that this is a memoryless process. It's a Poisson process, so there are certain frequency of switching from growth to shrinking and back. the certain rates of growth and shrinking. And there were actually very beautiful mathematical models describing details of this dynamic instability. So as soon as this discovery was made, they speculated. They really came up with this hypothesis, unsupported at the time with anything, that what is this dynamic instability for? They speculated that it can be used by the cell to stochastically assemble the mitotic spindle in the very simple process of the search and capture. So the idea is that each individual microtubule just repeatedly grow and shrink and grow in the random direction. And if by chance the microtubule grows in the direction of the kinetochore and bumps into it and makes a connection, here you are, you get your connection. And then you simply wait for hundreds of microtubules to randomly like that to connect to the kinetochores, and you're done. So obviously, stochastic part is now clear. It's all random. You don't have to regulate this process. There was a first model of the search and capture. The first model was actually proposed by Terrell Hill in the mid-80s, who was collaborating with Kirchner. But his paper was so unreadable. It was basically 150 pages, dense with equations, with very little words, which nobody could understand, that it's largely forgotten. And it was rediscovered in a very elegant paper by Hooley and Libor, who used basically calculus and elementary probability theory to compute the optimal regime of dynamic instability for the fast assembly of the spindle. And they estimated that with observed measured parameters of the instability, you need basically like 10 to 20 to 30 minutes to assemble the spindle. And this paper pretty much started this whole business of computational work on mitosis. Later, it became clear that this paper was incredibly oversimplified, and now pretty much nothing is left from the predictions of this paper. But it doesn't matter. I hope you will appreciate by the end of the talk that truly useful mathematical modeling in biology can only be done if you are not emotionally attached to your models. You have to be ready to let them go when the data overwhelms them. The usefulness is really temporary. The model is very useful if it guides experiment in certain historical range. So what I wanted to show you is first our latest effort to understand if this search and capture model can really lead to the assembly of the spindle. So the problem with these first papers was that they were basically back of the envelope estimates on a piece of paper. To what we worked over the couple of decades is to build a code to actually doesn't play. That's too bad. What should I do? I don't have those. Okay, let me try the following. If I do it here. No, it doesn't play. Well, never mind. So I'll have to tell you without actually looking at these movies in dynamics. So what we finally managed to do is to do the Monte Carlo simulation of the spindle assembly. We're simply mimicking the growth of hundreds of microtubules from the centrosomes. We have chromosome arms, which are dynamic and bendable. We have kinetochores on the centromeres of the sister chromatids. And we actually simply monitor on the computer when the microtubules reach the kinetochores and get glued to them. So we do many molecular details. So we have deformable kinetochores. We have molecular linkers from them to the plus ends of the microtubules, and we simply mimic this process and gathered a lot of statistics. It's really a harrowing computational simulation. To simulate 30 minutes of biological time, you need to do the simulation for 20 hours on a very powerful computer cluster, but like running those models for a few months, you can gather a lot of statistics. And so what came up from this work was the following interesting results. So basically, we simulated a bunch of the chromosomes. We started with close to 10 chromosomes and finally got to like 46, the number in a human cell. And one thing, we saw that the timing of assembly is fine. We can assemble the spindle in about 10 minutes, but the accuracy of assembly is really bad. So let me show you one of the previous slides. Here's what I mean by accuracy. So the cell wants the so-called amphitelic attachment. It wants the chromosome to be at the equator with two sister kinetochores bound to two opposite poles. So then when the cell is ready to divide, one chromatid will be pulled this way, another that way. But obviously, if you do this search and capture randomly, you'll probably have a lot of these errors, for example, syntelic attachment, meritelic attachment. So those are actually happening in live cells, and some of them are bad. They lead to developmental defects, like, for example, to Down syndrome, to cancer, to death. But fortunately, in a healthy person, the number of errors is stunningly small. There's one erroneously segregated chromosome per 10,000 divided cells in our body. So considering that there are tens of the chromosomes in one cell, it's basically roughly like 1 per million error, which is just stunning if you consider how this process is going. But the simulation, of course, in the absolutely optimized regime, predicts that only 50% of the attachments will be proper amphitelic attachments. There'll be a sizable number of all sorts of errors, which are inevitable with this stochastic features. We were actually surprised that the results are as good as they are. We just cannot improve them anymore. And search and capture model cannot be improved anymore because we have pretty much everything which is known, all the realistic 3D geometry there. There's nowhere to go. So what I mainly wanted to tell you about today is our latest effort in the last 2 years to understand what's going on here. So clearly, this is kind of the death call for this model. There has to be some new understanding. Now, one of the things I wanted to show you is one of the signatures of the stochastic process of the spindle assembly, is that if you record the events of the so-called biorientation, this is what biologists like to call this proper amphitelic attachment, is that these events are distributed roughly exponentially in time. So you have first to wait a few tens of seconds for the first connection to be established, and then, majority of connections established relatively rapidly, but a great part of them, more than half, are taking place slower and slower. And this is a very simple consequence of simple fact and probability theory, that you have n independently running random processes, and you want all of them to be completed. So you have to wait long time for the last one to happen, okay, because of the nature of this randomness and independence of these processes. So what we were missing all these decades before the last few years is actual biological data of what is actually happening with the centrosomes and the chromosomes, kinetochores, in space and in time in the cell. It was next to impossible to track kinetochores and centrosomes with high enough temporal and spatial resolution. And so finally, this problem was overcome. And what you see here is the computational rendering of experimental movie in one of the cells. So what you see are two centrosomes segregating. I'll play it again a few times. So they start closer to each other and then they start segregating. And those short segments, these are centromeres. So at the end, these tiny discs are kinetochores. So we have all 46 chromosomes, and we track each of the 92 kinetochores and 2 centrosomes with the resolution of at 5-second steps with spatial resolution of less than 0.2 microns. The latest advances in microscopy allowed to do that. So you see that, and you can see it in 3D from different angles of view. And it's not easy to just by looking at this to understand what's going on. So one thing is clear, that eventually all the chromosomes are at the equator of the spindle between the two spindle poles, and that there's this stretch of the centromere. So originally kinetochores are closer to each other. That's the color coding here. And then eventually the two sister kinetochores get stretched, because when they get attached properly to the pole, they get pulled by molecular motors in the opposing directions. But so the question is, what does the data like that tell us about the spindle assembly? So we started to analyze it computationally. And first, we simply plotted the trajectories of multiple kinetochores and saw how they move. So what you see here is the trajectory of all the chromosomes, which is basically the midpoint between two kinetochores. And the trajectories are color-coded, so that early part of the trajectory is a cold color and the late part is the hot color. And so what is clear is that originally all the, and so this is done in the frame of the spindle. So that's what we realized earlier. It's kind of useless to look at those movies in the framework, in the lab frame, because then everything kind of heaves, moves around the cell in such a complex way, it's becoming a mess. But if you move in the coordinate system, which is has the origin at the center of the spindle, and the z-axis is between two spindle poles, then everything becomes more clear. So in this coordinate system, at first, all the chromosomes move centripetally toward certain barrel-shaped surface. And then later, they start moving along this barrel-shaped surface. So that's what is happening. And that set us on the following path. So the beauty of this data is that we can measure tens and tens of different chromosome quantitative characteristics. I will mention just a few of them, but we can actually measure a lot of them. And we are now running all this stuff through some machine learning algorithms, trying to see if we miss something by eye. But so two most important ones are the interkinetochore distances. The stretch between this distance, again, is a signature of this amphitelic proper attachment. And the so-called spindle angle, the angle between kinetochore axis and spindle axis between two poles. Again, the decades of biological research show that the proper attachment, this angle should be 0 or pi, right? So that kinetochores are pulled in this direction, so they should be, the axis should be aligned with the axis of the spindle. And so that's when we plotted this two characteristics as function of time from number of cells tracked by this, we see that the angle indeed goes down to something very close to 0, and the kinetochore distance gets stretched. And so the definition of biorientation of proper attachment is if angle drops below a certain threshold of about 30-something degrees, and if the kinetochore stretch becoming greater than 0.9 microns. So now we can look at each individual chromosome and detect when does proper attachment take place, when does biorientation take place. So the first surprise that this data brought to us was the following. Again, to remind you, in the previous model, you have to wait for a long time for all the chromosomes to be attached properly. This is the prediction of the model, right? Exponential long tail. This is the actual data from one cell, but we have tens of cells all showing the same. So there's relatively narrow kind of Gaussian-like peak. Okay, so all the chromosomes are properly attached at once. The process is not stochastic, it's deterministic with some noise on top of it. Okay, so that is the first experimental discovery. Now then, when we started to look at individual trajectories of the chromosome in the framework of the spindle, we discovered the following things, that the trajectories can be by eye separated in two classes, good ones and bad ones. So the good ones kind of looks like that. The chromosome starts far from the spindle and then starts moving to the center of the spindle here. Meanwhile, poles of the spindle are segregated. And then when the chromosome bumps into a certain surface, again, this barrel-shaped surface, it will be clear later what the surface is. Then that's where the biorientation happens. Kinetochore stretch, angle drops to 0. Okay, this is good chromosome. There were always a few bad chromosomes in the cells chemically inhibited. I'm not going into the details of this biochemical inhibitions because I guess majority of you guys are in math and physics. I don't just want to overwhelm you. But so some of these bad chromosomes, here's what happens to them. They again move to the spindle, but they got to the spindle not close to the equator, but close to one of the poles. That will be important for the later. And then they didn't biorient. KTs didn't stretch, kinetochores didn't stretch, angle didn't go to 0. And instead, the whole chromosome went with one of the poles. So this is either syntelic or merotelic attachment. This is bad. So again, if we looked at the total data, we clearly saw that biorientation is achieved on this barrel-shaped surface. So these are the spindle poles, and this is the kind of density of the events of biorientation. Side view, view along the spindle axis. Okay. So chromosomes converge, bump into certain surface like that, right, and get attached immediately without any statistics. Then we started to build a model, mathematical model of what's happening. Now, when building this model, we took into account the following discovery from a few years ago, that kinetochores are not naked in their interactions with centrosomal microtubules. This is the discovery in this paper published in Journal of Cell Biology 3 years ago. There are short microtubules growing from the kinetochore with plus ends of the short microtubules, or the kinetochore minus ends are facing out, and there's this molecular motor called dynein on this minus ends. Now, dynein is the so-called minus end moving motor. It tries always to move to the minus end of the microtubules. So this motor here tries to go to the minus end of this long microtubule growing from the centrosome and tries to drag the kinetochore and the whole centrosome attached to it, tries to drag it to the microtubule pole, which is kind of counterintuitive a little bit, right? It means that the chromosome should collapse to the poles instead of staying in the equator. These are some experimental images that prove the existence of the short microtubules sticking out from the kinetochores. So our model was as follows. We started to mechanically model the centromere as two kinetochores connected by effective elastic spring. There were biophysical experiments showing that this is actually so. With the short microtubules sticking out of the kinetochore with motors at the end trying to move to minus ends of the long microtubules of the spindle. And we put into the model the detachment rate of the motors from microtubules, attachment rates, and we put in a very simple, very simple assumption that detachment could be dependent on the angle with which microtubule sticks out of the kinetochores. It's actually not very stringent attachment. It's as simple as kinetochores are kind of hemispheres. And we're saying that microtubules don't like to be kind of growing in that direction. They want to grow more or less normally from this hemisphere in this direction. So in that direction, they try to rotate into the normal direction, and they don't like to attach in this orientation. They like to attach in this orientation, or they detach faster in this unfavorable orientation. So we simply had certain polarity saying that microtubules, short microtubules and motors like to act when they are aligned with KT directions and don't like to act in perpendicular direction. And we wanted to see what this model would predict for how the chromosome behaves in a few different spindle parts. Now, what would be the difference between microtubules in different parts of the spindle? If you are at the equator, we know that these microtubules of opposite polarity overlap, and you have this network of bipolar microtubule network. It could be either uniform, right, or they could be bundled. So it could be like elastic cage built of these ribs of bundles of microtubules like that, or it could be just uniformly covered by microtubule surface. Away from the spindle, there are many long microtubules growing away from the spindle, and they of course overlap at certain angles. They're not anti-parallel like on the surface of the spindle. So we call it angled network like this. And then close to the spindle poles, it's a biased polarity. Majority of microtubules are growing in the same direction with the same polarity. So we examined how this contraption will behave on different networks like that. So mathematics of the model is very straightforward. It's the so-called Tug of War model, and we did two versions of the model. One, discrete stochastic models, when we are, in a way, serving Langevin equations together with random process and some mechanical equations of movement for each short microtubule, each molecular motor, and all the springs. And we balance the forces and torque. So we solve simultaneously mechanical equations for movement of the motors. Having what I want to emphasize that there are very little untested assumptions here, because molecular motors are researched really extensively in the last decades. And so mechanical equations are not in question. So the only important thing here is that we have a few, like order of 10 of the short microtubules, which is known from the microscopy. order of 10 long microtubules each of these chromosomes interacts with, and characteristic K on K off rate, which is of order of 1 per second. So that's pretty much it. All the other parameters are not that important. So there's this stochastic simulations. Then, because we are, after all, applied mathematicians, we just did a mean field limit of this stochastic model and derived a continuous equation. Basically, now instead of discrete microtubules, we have probability density for the distribution of the short microtubules over angles. And we have basically 4 of these densities that distinguish between 2 populations sticking out of different kinetochores, and each of these populations can stick to the microtubules leading the motors one way or the other way. Okay, and then there's interestingly, the equations are becoming integral ODEs, so it's not completely trivial. Integral ODEs because the action at different angles converges into the simple mechanical action of the chromosome. And so this model can be analyzed semi-analytically with the linear stability analysis, and it shows that what we have is the interesting polarity sorting phenomenon. You see, the problem here is that one kinetochore can be pulled in one direction and in the opposite direction, and same as sister kinetochore. So naively speaking, there will be complete chaos. Kinetochore will be just flipping every which way, orienting stochastically, nothing is going to happen. But what is happening is the following. Motors from one kinetochore gradually aggregate on microtubules of one polarity, motors from another kinetochore on microtubules of another polarity. It's basically this tug of war between similar motors is such that it's mechanically unstable to pull the object you attach to in 2 different directions. You better start pulling this object in one direction. That way, motors stay attached to the same microtubule for the long time. And then your sister has no other way but to start attaching to the microtubule of opposite polarity. So it's a very non-trivial thing, and this is the key for everything that happens. So let me show you some movies, computational movies that came up from simulation of the stochastic model. And they explain what's happening, what's the difference, for example, in the angled network away from the spindle and in the parallel network on the spindle. So in the angled network away from the spindle, here's what happens. That's not good. Sorry, let me see if I can play it here. No, sorry. Okay, I have to say it in words. I didn't test this talk on this computer. I tested it on another one. Anyway, so on the angled network, nothing good happened. The chromosome didn't get stretched much. It was wobbling all the time and kind of walking over this network. So basically, if the microtubules of opposite polarity are not parallel to each other, then the biorientation was not happening. But beautifully, what was happening was that chromosome was moving toward the spindle this way. Now, on the surface of the spindle, it was nice. The chromosome was stretched, but very often in the wrong direction. So motors from one kinetochore could bind one polarity microtubule going this way, from another that way. And so the chromosome ended up in the direction kind of across the spindle. So it's not quite a proper attachment. then what happened was that the perfect results, I'm pretty sure it's not going to work either, yeah. So the proper results happened when chromosomes started to interact with bundles, discrete bundles of the microtubules when microtubules are close together, because then opposing polarity microtubules stretched the chromosome in the direction parallel to pole-pole axis. Now, these are the results of these stochastic simulations, which show a fraction of bioriented chromosomes growing as a function of time. And you see when they move on parallel bundles, 100% of the chromosomes got bioriented, and they got bioriented over about 40 seconds. And this time, 40 seconds, comes from a very simple thing, that each motor attaches and detaches on the scale of second, and altogether, you need a few tenths of these processes of attachment detachment to get the problem solved. Okay? If you try to do it on the uniform microtubule surface, only 50% of chromosomes get bioriented, lots of errors, and on angle, very, very, very few get oriented. So the model, right, so here I'm already listing the predictions of the model. So the prediction number one, chromosomes should biorient when they hit the spindle. So this barrel-shaped surface should be the spindle. That's where the antiparallel microtubule bundles are. Second prediction, the spindle surface should be made of microtubule bundles, not of uniform microtubule network. Third prediction, after hitting the spindle, it takes tens of seconds to achieve orientation. Now, prediction number four, it is important to hit the spindle near the equator. So those are trajectories predicted by the model. And say the chromosome stays near the center if it hits the center of the spindle. That's where anti-parallel microtubules are. Hit the spindle closer to the pole and you move to the pole. Remember this experimental data, hit the spindle near the pole, you will move with the pole and no good will come out. So these four theoretical predictions we then tested in the experiments, and they were very beautifully confirmed. One is this, we can start imaging microtubules and crosslinkers that crosslink them, and at the same time recording the points in space and time where chromosomes get bioriented. And see, this is the outline of the spindle, and these are the biorientation events. So indeed, hit the spindle, get bioriented. Second, that the surface has to be made of bundles. And indeed, this is true. So when crosslinkers, RC1s, crosslinkers that crosslink antiparallel microtubules at the equator of microtubule were imaged, you have this, you have those ribs that are at a few microns distance from each other instead of the continuous surface. And you can also kind of see this discrete ribs along which chromosomes move after hitting the spindle. Then the most dramatic data was the following. At a very high spatial resolution, you can image individual microtubule bundles of the spindle and two sister kinetochores. And you see, this is the moment when the chromosome is hitting the bundle of the spindle. There's already somebody sitting there. So after the hit takes place, see the angle so far is wrong, but the hit starts and then angle get aligned and the interkinetochore distance get aligned, and we got statistics of this time. It happens roughly over a minute. In the experiment, we predict about 40 seconds, so it's pretty interesting and dramatic confirmation of the model result. And finally, the fourth prediction, hit near equator. That's where we started this data science, like this big data, which is so fashionable these days. Of course, it's kind of useless to look at this a few thousand trajectories from tens of cells we have by eyes. So we started to feed them into the computer and make the computer to do some advanced clustering. So what we do is we look at the trajectories of individual sister kinetochores over time in the spindle coordinate frame and start measuring certain quantitative characteristics of the spindle. So for example, one very interesting characteristic is early trembling. So when early on, when the chromosome is not even interacting with the spindle yet, there's certain noise here. So it's chromosome starts trembling. So we quantify it and say that it's weak trembling or strong trembling. Of course, interkinetochore distances, the spindle angle. We also measure late oscillations of the chromosome at the equator. That's normally the signature of proper attachment. And of course, where the chromosome hit the spindle. So then we convert it into binary data, assemble the data for each trajectory, do clustering of the trajectories. And then this clustering procedure tells us very simple answer, that there are two different types of the trajectories. And I already actually showed you these two different types of trajectories, good ones and bad ones. So what I showed you here was the extreme examples of this behavior. So this is extreme example of bad behavior. This is extreme example of good behavior. But clustering shows the actual data without bias. So all of this is done in unsupervised way. So cluster 1 is bad, cluster 2 is good. So see whether chromosome ends far or close from the center of the spindle, from the equator. These ones are pretty much on the equator. These ones are pretty much on the pole. So this is stark difference between good and bad. Oscillation, late oscillation is also a very good criterion. Good chromosomes oscillate a lot, bad chromosomes just sit there motionless. Early trembling, amazingly, is a very good distinction between good and bad. Good chromosomes should tremble a lot very early on. So this is the only early indicator of the chromosome fate. But there is one. So it's interesting that there's something very early on that tells whether chromosome will end up correct or wrong. Those are not very interesting. And finally, this is the confirmation of the experimental prediction. Closer to the pole is bad. You belong then to bad cluster. Okay, so that was the last confirmation of the model prediction. So this unexpected prediction from the data that early trembling is good. Unfortunately, again, I cannot show you this movie, but if you could see them, you would see that early on, when chromosomes interact with this angled network, they tremble a lot. So we think that this trembling is very simple. It's interaction of the short microtubules with the long microtubules. And because the number of the short microtubules is of order 10, the mechanical noise is very, very large. So it's simply this mechanical noise that makes chromosome tremble. Absence of trembling means that there are some other types of interactions with microtubules. Probably the kinetochore itself, naked kinetochore, interact with long microtubules and just smoothly slides along it. And that's bad for chromosome because this stochastic tug of war, this polarity sorting, will not translate into the proper attachment of chromosome at the end. So that's pretty much the end of my talk. So the big kind of philosophical conclusion here is that we are moving away from either of the initial concepts. One is search and capture assembly from two asters and individual chromosomes. And we are not endorsing either the centrosome-less assembly by short microtubules from kinetochores making the spindle on their own. But we are saying that it's a hybrid. It's interaction of the astral microtubules with the short microtubules, and then interaction of the spindle, central spindle with this short microtubules network that make the spindle rapidly and accurate. And so, of course, there are many interesting puzzles remain. One is what I didn't talk to you much about is this. Remember, I mentioned this convergence, centripetal convergence of chromosome to the spindle, right? So this is very rapid and relatively synchronous. And again, we are not sure yet how this is happening. In principle, we can address this problem with the same model that we already have. We didn't start yet, but I already anticipate some difficulties. So we're still missing some crucial things here. Even bigger problem is the proper end-on attachment of microtubule to the kinetochore in the end. In the end, you want long microtubules from the pole continuously go to the kinetochore and attach to it. And what I showed you is kind of only preliminary step here. The last step, we're not totally sure yet what's happening. So I hope you appreciated a few things here. One is that combining relatively simple computational models with serious quantitative data can work, can help in uncovering the secrets of the most fundamental process in cell biology, cell division. The second one is that we are not really moving away from this philosophical concept of very stochastic, very noisy process, somehow converging in the end in extremely deterministic and accurate result. And so, to my mind, this is really the main general conceptual problem, very exciting problem for mathematical biologists in biology. How, with tens and hundreds of very stochastic, noisy players with many errors in the middle, in the end, we get this very rapid and faithful result. And there are, of course, hundreds of molecular machines in the cell, different from the spindle, but many of them are characterized by this feature. And so this is our greatest modern challenge for mathematicians to help biologists to understand the design principles behind it. So again, those are the people who did this project. And thank you very much for being here in the late afternoon in Spain or wherever you are. Thank you for listening. And I am ready to answer any questions if there are any.
Thanks a lot, Alex. Thanks a lot.
Sorry, I don't see anything in the chat.
Oh, okay. So I would like to express my gratitude because I think this was a very beautiful and interesting talk.
Thank you.
And probably we have time to have a few questions. So if you would like to pose some questions to Alex, please state it in the chat and we will give you the time.
To open the question, I would like to thank you, Alex, for the nice talk. I would like to ask about the orientation of the microtubules. If the cost of the energy, because if everything or most of the processes is well, stochastic in some sense, the cost of the energy to there is not some additional biochemical potential or something like that could orientate the microtubules, and in order that minimize the cost of the energy because I imagine the cost of the energy through this kind of microtubules is high for
Yes, so that's a great question actually. So for decades, many people, including myself, were completely ignoring this energy demands on the cell and those processes. The thinking was that so the ongoing metabolism of quiescent cell consumes enormous amount of energy every second. And so it was thought that the process of cell division, for example, or cell movement is just like 1% of the total energy balance of the cell. And then who cares? Those processes are so important that, of course, cell can allocate 1% to them. But lately, it's becoming clear that it's not 1%. This is tens of percent. And exactly as you said, each of the cycles of the microtubules consumes a lot of energy, like each dimer of tubulin consumes one GTP, like one smallest energy currency of the cell. All this molecular motors spend energy, the kinases, phosphatases, activating, deactivating motors. So it's enormous energy. Unfortunately, we have very little knowledge of what the cell does about it. Just, keep producing lots of this energy and allocating it to mitosis, maybe. It's becoming pretty clear that if you start starving cell of energy, it will delay division. So those are interesting questions, but we have almost no idea what about anything interesting to say in this regard.
Okay, I would like to pose another question myself. So I would expect that there is nothing fundamentally different about these dynamics in cancer cells, right?
Yeah, so that's a great question. Like the main processes are all the same, but there are, of course, very important differences. And like one of my favorite topics is this. Many types of the cancer cells, instead of 2 centrosomes, have more, 3 or 4, and so on, right? But amazingly, they still assemble proper spindles. So those centrosomes have property to cluster into 2 clusters. And so each cluster is becoming a new spindle core. But in the process, they make a lot of errors. And that's one of the reasons that cancer wreaks such havoc on our organism, that those cells, when they divide, there are many errors, so they are genetically different from normal cells in our body. And so even cells with 2 centrosomes in cancer, they divide faster, but with more errors. And so that is what is interesting. So the mechanics is all the same as I described, but the regulation is all different in cancer cells. And it's very interesting to compare this mitosis with many errors in cancer with mitosis with very few errors in normal cells.
I see. And is it, let's say, can it be envisioned that this information will be of use to design some specific kinds of chemotherapy, or is a pretty high order, so to say?
Yeah, also really excellent question. So I'm not a big expert there, but I know that in the past, for example, there was this highly publicized attempt. So there are some pharmacological drugs. One is called monostrol, which disrupts the central spindle. So it inhibits one of these molecular motors I mentioned, which makes this central spindle kind of disheveled. And that very often causes the cell to abort the process of mitosis and the process of division. And from what I know, this drug is still a part of some of the cocktails of anti-cancer drugs. And there are other attempts like that. So largely speaking, the practical applications are still a little bit murky. But, yeah, the people are thinking about this. because that's, of course, at the very core of the pathway to cancer, the abnormal mitosis.
Oh, that's quite interesting. I hope they succeed.
Yeah.
Another question related to your model is the force you propose is in some sense attractive because you propose some the distance and then you put 1 minus the force over the modulus, more or less, if I remember. Then why did this force? Why this kind of force? Is proportional to the is proportional to the is the direction? It's a direction, in fact, between two, but is a linear.
Yeah, so I'm sorry, yeah, I went too fast about it, but it's basically it comes from well-accepted models of molecular motors. So generic molecular motor, it tries to move to the end of the microtubule with certain speed, like so let's say V0. And then if this motor is moving slower, then it tries to push and moves faster. So it moves against certain force. And these so-called force-velocity relations were measured many times. And they are some nonlinear functions, but they are normally monotonically, so velocity is normally monotonically decreasing function of force and vice versa. So for simplicity, many models simply use linear function. Velocity is linear function of force and vice versa. So that's where it's coming from. There was a couple of rigorous studies showing that those nonlinearities don't really matter. And it's much easier numerically to work with linear functions. There's additional question, what does it mean chromosome is bi-oriented? So it's as simple as it's amphithelically attached. So two sister chromatids, two sister kinetochores attached to microtubules coming from the opposite poles and pulled in opposite direction. Sorry about this terminology, like this is biology, they're full of exotic terms.
All right. So if you don't have any more questions, I think we can conclude with the seminar. Thank you again, and everybody for being here.
Thank you.
I hope you enjoyed it.
Nice talking to you.
Nice talking to you.
We hope you finally came to Granada in the next future.
I hope so. I love Spain.
Yeah.
Bye.
I hope so. So let me briefly remind you that next week we are going to have Anna Marciniak. Same day, same hour.
Okay. Bye.